let me say something about that right now. So hemoglobin is 4 protein chains, all right, 2 what are called, this is the Greek letter alpha chains, plus 2 what are called beta chains. I'll show you more about what that is. Basically, there's 4 blobs of protein there. It's like saying there's 2 apples and 2 oranges stuck together. They're similar but not identical. Let me write that a little more neatly. That's a Greek letter alpha, and that's a beta chain. Plus 4 heme molecules. That's the red part that Levi was just asking about. And oxygen binds here. So the whole thing is about 4,400 atoms, give or take. And so it actually contains, so there's the heme part, which is proteins, made of a bunch of amino acids stuck together. We're going to talk about that in extreme detail in a few minutes. I'm sorry, the globin part. The protein chains are, the alpha chains and beta chains are the protein part. They also have what's called a prosthetic group, like a prosthetic limb, something that helps them out. That's the heme molecule, that flat red part. I'll show you more in a second. The spidery red part, that's actually where oxygen binds. That's a separate thing. It's not made of amino acids. It's made of carbon, hydrogen, nitrogen atoms stuck together in a particular shape that oxygen binds to particularly well. And so the whole thing together is about 4,400 atoms. Let me go back to the structure and show you a little bit more of what I mean by that, all those different pieces. So, and again, these pictures are in your handout. So let's turn the spin off for just one second. So back to the hemoglobin. Another way to look at the hemoglobin is the yellow ghosty stuff. That's all the atoms of the protein. The red spidery things, those are those heme groups. Again, they're not made of amino acids. They're a separate molecule that is bound to the protein that binds oxygen. So it's a 3-way thing. So the protein holds the heme holds the oxygen. And there are 4 heme things, because 4 oxygen molecules bind. And so you can see when you add oxygen, poof, they bind to the heme, they unbind, bind. Before we go further, sort of orientation questions, what are we looking at? Oxygen, deoxy, heme, that sort of business before we go further. Okay. So you could also look at it in terms of the backbones, all right? And here again, you can spin it around. That sort of makes the protein just a little less sort of in the way. And again, you can have oxygen and not oxygen, all right? What I want to zoom in on is the following. So here's hemoglobin again. And I remember I said it was 4 subunits. So that's the 4 subunits. So if you look at these guys, one functional unit of hemoglobin contains several pieces. 4 protein chains, 2 alpha and 2 beta. Those are the blue, yellow, green, and light blue. So if you think about it, it's sort of 4 blobs stuck together. If you like I said, 2 oranges and 2 apples. 2 alpha chain, 2 of one flavor, alpha kind of a chain, and 2 of the beta chains packed together. And each one of those blobs, the blue blob, the green, the yellow, or the light blue, has a red thing stuck in it. That red thing is that heme again, where the oxygen binds. So it's 4 pieces stuck together, each of which has a heme unit in it. That's where the oxygen binds. Right? And what I want to focus in on, because to simplify things, I want to zoom in on one piece. So again, that's another way to look at it. Those are the 4 chains, and the big fat red things again are where the oxygen binds. All right. So what I want to do now is zoom in. So take this thing where there's 4 subunits, and I want to zoom in on one of them, because just to simplify things, let's cut it down. That's one of those blobs, one what they call beta globin subunit. Okay. So again, there are 2 in any hemoglobin molecule. There are 2 alphas and 2 betas. So this is a typical protein. I want to talk about this in some detail. It has this compact blob shape. It was born as a long chain of amino acids. How does it have that compact blob shape? We talked about that a little bit on Monday, and I want to do more specifically now. All right, so that's to zoom in on what we want to talk about. Let me go back to the notes, and then we'll come back to the pictures. All right, so I want to zoom in on, so for example, one molecule of beta globin. That is one so-called beta chain. Okay, we could have looked at the alpha, we'll look at the beta. And I want to talk about first, and this is in your handout too, levels of protein structure. There are several ways to look at a, so this is ways to look at a protein. Okay. And they're numbered sequentially. People tend to think of them as one before the other, and they actually aren't. It's more like ways to look at a car. You can look at a car by what color is it. You can look at a car by whether it has four-wheel drive or not. You can look at a car by what its gas mileage is, right? They're all independent things. They're just different ways. You learn different things about a car by asking about its gas mileage. You learn different things about its car by asking about its color. Likewise, about a protein, there's different kinds of structure, and you learn different things about them. And so the first one I'm going to talk about is what's called primary or first-degree structure. And again, for reasons that aren't quite clear to me, these things are numbered, and they really sound like they're in order, like proteins must form this way. It turns out they don't. It's really just different ways to look at them. So what is primary structure? It is the sequence of amino acids in the protein. Okay? In the protein chain. And in the case of beta globin, if you look on your today's information packed handout, what page is it? Page 5, it shows you the amino acid structure. So you tell me, what's the first amino acid in beta globin at the amino terminus? On page 5, the very first amino acid in that list. Yeah. VAL, which is valine. That's right. So beta globin, it has its amino terminus. Then it goes val. What's next? Is it his, which is histidine. His, da 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 da. And what's the last amino acid? Histidine also. And then the COO minus group. All right. And there's, if I remember right, 146 amino acids. So that's its primary structure, all right? So what good is that, all right? That actually tells you exactly what atoms are present. If you wanted, it would take you an enormous amount of time. I would never ask you to do this. It would be an extreme patience tester. You could actually write out the complete chemical structure of beta globin, right? Because you can look up, what's the structure of valine? Peptide bonded to histidine. What's the structure of histidine? Write that out. Peptide bonded to the next one. Peptide bonded to the next one. You could actually write out every single atom in beta globin if you wanted in that stretched out form. Like I said, it would be an enormous pain and wouldn't be much point to it, but you could. So the primary structure says, this is a kit of parts in exactly what order we put them together. Right? So that's one way to look at a protein. Like one way to look at a car is it's, what color is it? This tells you exactly what amino acid is it at which position. How many are they? Exactly what order are they? And it is this sequence which determines the structure. If you remember when we did those wire things, everybody's protein was different because everyone's amino acid sequence was different. Well, and if somebody asked the question about sequence and structure in here, that particular sequence will always form beta globin and only beta globin. And it is that sequence that makes it beta globin that gives it that shape. And like you experimented with that protein folding program, what you saw is you type out that linear sequence of amino acids and it folds up automatically. And this will fold automatically into the proper shape. So that's number one. Questions about that before we talk about the next level? Okay. That one's relatively straightforward. The next one is shown a little further along in the page as secondary, second degree structure. And that's what they call major structural elements. So the major structural elements of the protein, and they're formed by H bonds between parts of the backbone. And this is a sort of an exception that in here we're not even caring about the side chains, just backbone stuff. And there's 4 types. There are what's called an alpha helix, which is a coiled backbone. There's what's called a beta sheet, which is sort of parallel backbone segments. There are turns, like that, and there's random coil, that is miscellaneous. Okay, and there are a variety of confusing things in this part of the lecture, right? People think that primary structure forms, then secondary, then there's another one called tertiary. It's not that way. It's just another way to look at it. The other thing is we have 2 alphas and betas. We have alpha and beta globin, which are names for 2 proteins. And we have alpha helix and beta sheet. They're actually not related. It's the first one they found and the second one they found. So I want to show you some pictures of what an alpha helix looks like to give you an idea what I mean. I'll let people write this down for a second. So secondary structure is sort of like, in a way, simplifying you to an X-ray. Where are your bones at, right? It doesn't tell you all the details of your, what your face looks like and your exact physical features, but it gives you a general idea of your form. And that's what these things are, sort of major structural elements in you, sort of like the skeleton of the protein. So if you look on the handout, there's some pictures of what I'll show you on the computer. So again, this is beta globin's backbone in blue, and the side chains in yellow. It's a particularly confusing way to look at it, but just to make it clear, there's the spirally backbone in blue, and then the side chains kind of hang off. That's a sort of structured protein backbone side chain business. Right? And then we can look at the backbone of just beta globin. And what you see, let me turn the spin off so it's a little easier. At the moment, it looks sort of, knotted up, kind of like what you guys made in lecture. But if you look, there are certain regions. Let me see if I can find one. Yeah, if you look, find a good one. Oh yeah, there we go. There's a good one. If you look, watch down here as I spin this guy around. What you're seeing is the backbone. If you look, you can even look right down it. It's coiled up. That's a little region of alpha helix. In fact, all the red regions of this protein are bits of alpha helix. There's another one. If you watch over here as I squeedle it around, you can see it's coiled up like that. So what those things form are sort of structural rods in the protein. They give the protein some rigidity. The backbone's coiled up tight like that. It gives it, like I said, a sort of stiffness to give it its particular shape, because its shape is what's going to determine its function. So let me show you one example. If you zoom in, there's one alpha helix. And you can see, like I say, it's coiled like a spiral. The backbone's coiled like a spiral staircase. And it's actually, you can look right down the middle. It's coiled like that. So what keeps it coiled like that? Why doesn't it form some other shape? Why doesn't it come uncoiled? Because of the coolness of hydrogen bonds is the answer. And it turns out that's the whole thing. And here I can... What you got... This is the backbone atoms. All those dotted lines are hydrogen bonds. Between blue, that's a nitrogen. And there's a hydrogen here that's invisible because they don't show hydrogen. And that is an oxygen right there. And there's this regularly repeating pattern of hydrogen bonds that locks that thing. So it keeps that coil. There's sort of rungs of the spiral. Well, not rungs aren't the right way, but they're braces along this spiral that keep it in that spirally shape. So hydrogen bonds in the backbone make this rigid, and the beta sheet works the same way, a whole bunch of hydrogen bonds in the backbone. Doesn't even matter what the side chains are. It gets locked up into this particular shape. And that, again, forms like an arm bone or leg bone. It gives the protein some structure. Questions about that one? Yeah. So the question was, are hemes, those things, those red things, found in all proteins or just hemoglobin? The answer is they're found in hemoglobin and myoglobin, a couple of related proteins, but not all proteins, because not all proteins carry oxygen. Only ones that carry oxygen. Yeah?
All the backbones are the same. How do you differentiate between an alpha helix and
So if this is only the backbone, how does it know to be an alpha helix? Why isn't everything an alpha helix? That's actually a really good question. It turns out certain amino acids, their side chains help make this work. They don't form the actual bonds of the alpha helix, but they make it easier to form. So it turns out that certain kinds of amino acids favor alpha helixes, it turns out. That's a good question. All right.